So I'm a computer scientist involved in brain imaging for 15 years or so. So I'm going to tell you what we try to do with computational tools on functional images, functional brain images, to study how stimuli, for instance, are encoded in brain activity. Well, there can be a culture clash where I use terms that are not familiar to you or concepts that are not familiar to you, so please raise a hand. I'm not here to sell anything, I'm just here for you. So if you need some more explanations, just raise a hand and I'll be happy to provide more explanations and hopefully be clear. All right. In this presentation, so there will be 3 parts, as in any good presentation. The first one will be just definitions of what I call an encoding models, or what the community called encoding models. So how we can, the type of models that are used to analyze functional images of the brain. And then I will go a bit more into detail. I will give an overview of the main sensory representations that have been studied by the community in the recent years, and the crucial question of model comparison. And then I will focus on validation, because to me, new ideas, new technologies should always come with good validation so that we make sure together that we are on the right track. Okay. So this is my framework. So I'll be talking mostly about functional magnetic resonance imaging, and I will assume that you kind of know what fMRI is, but well, this will be your crash course on fMRI if you need it. So typically in cognitive neuroscience, you may want to assess some theory of how the brain works. So some cognitive theories. This doesn't work. And so to probe such a theory, you will pick a certain set of stimuli, of things that, or tasks that you want to be performed by a subject during an experiment, and you organize them in a so-called experimental paradigm. It's just a sequence of tasks performed by the subjects during an acquisition. So you take the fresh brain of a 20-year student and you put him or her in the scanner, and you get these brain scans while the subject is performing the tasks. And then at the time of data analysis, you can try to relate the fMRI data that relate to the stimuli. And encoding models go from stimuli to brain images. It's a model that estimates the effect of a certain cognitive task on brain activity. And decoding is the converse operation in which you input a brain image and you try to predict what task was performed, what stimulus was presented. Okay? So actually today, I won't have time only to cover mostly encoding, but we can talk about decoding later. By the way, I'll be leaving tomorrow at noon before Brexit. So, well, if you have any question or want to discuss, we can discuss tonight, of course, or tomorrow morning. Maybe before we go further, you may know or not BOLD, functional magnetic resonance imaging. BOLD means blood oxygen level dependent, so it's just a modality that we physically use to physically measure the brain response to the stimuli, so the neural response to stimuli. It's an indirect measure that actually does not measure neural activity, but the blood response, the blood oxygenation changes that follow neural activity. We believe it's a relatively good marker of neural activity, but it's imperfect, of course. So we could use other modalities like EEG and MEG if we work on humans, or direct electric recordings if we work on animals. So I'll be focusing on BOLD for simplicity here, because I think it's quite advantageous overall to study what I call brain encoding. So the advantages of BOLD imaging is that you have full brain coverage. So you can map the responses in all brain regions, and this with a decent spatial resolution, about 2 millimeters isotropic, which is okay to understand what the brain regions are doing. Of course, it's not sufficient to map cortical columns, for instance. So we don't have an excellent resolution. We cannot really map the detail of functional architecture. Just we are getting close to it, but not to that level. Also, maybe the big disadvantage of the bold imaging is the temporal resolution, because we are sampling a blood response that takes 5 seconds to occur after neural activity, and also induces a blurring of the response in time. So we have a poor time resolution in the data. We can only have of the order of 2 seconds. And I think I will discuss it more later, but we don't have a real access to neural mechanisms. We just kind of see the decrease of local activity of the sets of neurons, but this is very far from mechanisms because we don't have the resolution, because the measurement is too indirect. Okay? So let's go a bit further now. So remember, we put the subject in the scanner, and we presented some images to the participant. And so the images were processed by the participant. So you can think of different kinds of mathematical operations that are implemented by the neurons in the cortex. And so the neurons activated depending on their properties. And we observe through a complex metabolic pathway the bold response. If we formalize this, we start with a stimulus x. So x is a vector of value that represents your input. Could be a natural image that you presented to the participant. Then from that, we'll create a stimulus representation, phi of x. This is really the core concept today. So the pixel values of an image may not be very interesting or very relevant to understand brain activity. We rather want to extract some features. Could be a color histogram, could be measures of the contrast in different parts of the image, etc. And then in a given brain locations, phi of x is high dimensional typically, because you cannot represent a natural image with just a few numbers. You typically need a relatively large collection of numbers to represent a complex stimulus. So phi of x is high dimensional, but in a given brain location, you will have some coefficients beta that tell you how strongly the neurons are sensitive to the different dimensions of the representation. So you will measure phi of x dot beta. And actually, you don't measure that. What you observe is a fMRI activity y, which is this thing, the neural activity, but convolved in time with a hemodynamic filter, which is just due to the blood response, the blood oxygenation changes, which are represented by the temporal filter h. So think of this as events occurring in time. You need to convolve their time occurrence with this hemodynamic filter. Then on top of it, you've got some nuisance parameters like respiratory effect, motion of the head, etc., plus some noise. And this is the first model of the bold signal that you observed. There's nothing new here. This is a very classical thing. Now, you've got x and you've got y. Okay? You've got the stimulus you presented and you've got the bold signal you measured. So how are you going to analyze this? Well, first of all, you will create the representation phi of x that you fancy, that you want. It's your choice as a modeler, so to decide which features of the input may be relevant to explain brain activity. And then the most common way, once you have created this phi of x, the most common way to proceed is to take the bold signal y and from it to essentially deconvolve the signal. So you will essentially extract the response to the presentation of stimulus x. So an estimate of the neural response for this given trial. Okay? And then, so this is the first step, and then second step, from this you will compute beta, which is actually your parameter of interest. So beta will mean in a certain brain locations, the neurons are more sensitive to, say, the color information in the image, or whether in the image there was a face represented or not, etc. So this information of the feature selectivity of the neurons is the beta vector. Does that make sense? Yeah. And then one other possibility is also to estimate all the parameters jointly in a big minimization problem. I will just now make this a bit less abstract by showing how it works in practice. So you've got your fMRI data, which I called Y in my previous slide, and you create typically a model, a temporal model of your experiment. So this is called a design matrix. So you know when, across time, when you presented the different stimuli. And in typical fMRI experiments, you would say, in that case, show images of famous faces, scrambled faces, or unknown faces. So 3 types of stimuli, which are different events in time. And in most fMRI experiments, you take all the events of one type and you make one regressor to capture the response to this condition, okay? And plus some regressors of no interest, which are here. And so typically in a traditional fMRI analysis, you would get the activation for famous versus unknown faces, for instance, and compute the difference in response to these 2 conditions, or scrambled versus unknown face, typically. But when we do an encoding analysis, we do things a bit differently. We develop the design into this kind of matrix in which now each column is for one single event. So this is what I called previously the deconvolution step, okay? I told you that what you observed was a neural response to each event convolved with the hemodynamic response. Well, you've got it here. There was some event in time, and here you see the result of the convolution, assuming that the hemodynamic response is known. You see the result of this deconvolution, and by computing the correlation of this regressor with Y, you will get what I called previously phi of x times beta. Okay? And this is very classical. It's a relatively standard fMRI data analysis. So more in detail. For each of the columns of the design matrix, you will obtain one brain image that shows you the amount of activity for each trial of your experiment, okay? Each column being a trial here. These images are ugly, they are noisy because you've got very little signal to capture the effect. So typically you will have a rather noisy snapshot of brain activity. And then, so this was the deconvolution step. And then in the encoding analysis proper, you take the stimulus that was presented for the first image, which was like a face image, maybe a famous face image, okay? And you say, okay, I create some representation of this stimulus, which could be the contrast of the image or some global information about the face. And you try to, you will relate this phi of x to the activity that was recorded for trial 0, trial 1, and so on, trial 11. And probably you need many more trials to make a comprehensive analysis, but that's the idea. Is that clear? Yeah? Okay, so this is how people do in reality when they do this type of analysis. I won't go much into details here. There's also the possibility, so in the previous slide, I assumed that the hemodynamic response H was known. This may not be the case. If it's not the case and you have a lot of fMRI data, you can also try and estimate it from the data. So you can estimate jointly the H and the beta. This is a rather complex thing technically, but Then you obtain some estimates per voxel of the hemodynamic filter. So here on the data set, we did that, and we obtained a family of H filters, one per voxel. So you see that essentially there are some variations around a standard model that essentially peaks in 5 seconds and then has an undershoot. When you have a lot of data, it is advantageous to learn the hemodynamic filter from the data rather than imposing a predefined hemodynamic filter. You can see that it increases the performance of the encoding model. Okay? If you want to get more details, I give you the reference here. But I just said that the performance of the encoding model is better, so I need to define the performance of the encoding model. Remember that the encoding model, it's interesting because it's a generative model. It's a model that essentially is based on how the data are generated. So once you have estimated the model on your data, that is, you have learned the beta coefficient, you can take then new stimuli and then simulate how the brain would respond to this new stimuli. So you can essentially now guess the Y that would come if you put a new X into the model. Okay? Because you know everything to do that. So this means that you can use some validation measures like cross-validation. You take some data, stimulus, fMRI images to train the model, to learn the beta, and then you take another fold of the data to another subset of the data to test the model. So based on X, you simulate the Y, and you see how far your simulated Y is from the true Y. And this is measured by the R2, the coefficient of determination, the R2 coefficient, which measures the difference between the true fMRI activity observed in the test set versus the one that you estimated from your simulation, compared to simply the variance of the Y in the test set. Okay, and this number is smaller than 1. If it's 0, it means basically that you have learned nothing. You are not doing any better than just predicting the mean of the test set. So you haven't learned the variations in your signal. And if it's a very good model, it gets close to 1, because if it's very good, this goes to 0, so the R square goes to 1. Okay? This R square is measured in each voxel. So for each brain location, you will be able to say, oh, my encoding model works or doesn't work, which is very important for fMRI because we have good high resolution. So we will be able to say that this brain region is well explained by the encoding model, while this other brain region is not. There are also some other possibilities, but let's move forward. So to summarize a bit the idea, or put it with a different picture. You've got some stimuli that are presented in an experiment. This is called the input space. And you can see each of the stimuli as a certain vector in a high-dimensional space, the space of pixel values. So the number of dimensions would be the number of pixels in the image. But typically, this X representation, where you just enumerate the pixel values, is not interesting. It's not meaningful. Okay? Probably you want to extract some features from the image, like the presence of some objects, the color information, the amount of contrast in the image. And by doing this, you create a feature space. And so you encode your image in a certain feature, different feature dimensions. This is phi of x. And the mapping from x to phi of x can be nonlinear. And then the final thing is the brain activation that you recorded with fMRI, which means that for each sample, you've got a vector of brain activity, and it's a vector in the voxel space where each voxel is a brain location. And so you can characterize each sample by the activity it elicited in each voxel. So phi of x is high dimensional, y is high dimensional, and y is very noisy, obviously, because measuring brain activity is hard. So we don't do anything fancy here. We just use a linear mapping to relate the feature space to the brain activity. Actually, if you think of it, since you need to feed the model for each brain location, and there are like hundreds of thousands of voxels in a brain, and you are fitting a high-dimensional model because phi of x is high-dimensional, you need a very efficient model to associate phi of x with y. So that's why from the beginning, I implicitly assumed that it was linear by saying, essentially, we're going to learn a beta vector that relates the feature space to the voxel space. Okay? And by the way, decoding is not much different. When you do brain activity decoding, you just do the same operations, just you reverse that arrow, okay? Instead of mapping the feature dimensions of the input to the voxel domain activation, you just predict the feature characteristics from the voxel, from the brain activity, so from the values in the different voxels, and that's it, okay? So actually, the encoding and decoding are just, two sides of the same coin. Okay? And personally, I can only learn by doing things. So if you would like to understand more and try, we provide some examples in the NiLearn library. It's an open source library that you can just download now if you like. Install and play with the examples. There are examples in which we show you how it works. So we take data from a real experiment in which some visual things were presented in a 10 by 10 screen, so a 100 pixel screen. And you've got brain activity. And you can, so I will focus here on the encoding information. So for 4 different voxels of the image domain, you can see here, the 4 voxels are here. You can see the pixel in the image domain that elicited activation in that voxel. So these are the beta coefficients for each of the voxels in the brain. Okay? And what you see in color here is the set of R-square value in each voxel, how well you can predict the activity of that voxel in a given in unseen fMRI data given your model. And you see obviously that you can predict brain activity only in the visual cortex, in the primary visual cortex. Decoding is the converse operation. So essentially, you can see how well each pixel can be decoded from a brain activity. And for instance, for this pixel here, you can see the pattern of a voxel in the brain that predict the activity, what happened in this pixel in terms of stimuli. So for a nearby pixel, you would see some neighboring voxels involved in the prediction. Okay? So play with it. Okay, let's go on. I will go a bit deeper now into sensory representations and the question of model comparison. I think there are 2 key intuitions. At least these are my intuitions when I work on that. The intuition first is that for many sensory systems, there is a correspondence between the physical stimulus parameters and the brain representations. We can call that functional gradients typically. So the best known case is that of retinotopy. Okay, you've got the visual field can be modeled at the 2-dimensional structure. Okay, and it maps onto the 2-dimensional surface of the cortex. Okay, I will assume here that the cortex is a surface, which is not exactly the case. but forgetting the depth of the cortex, we can consider it as a surface. And so the two-dimensional visual field maps to the two-dimensional cortical surface, and it maps smoothly. So it's a diffeomorphic relationship in which your sensory space, so here the locations in the visual fields, are mapped to actually to the V1 regions or the retinotopic regions. Okay, so the general principle is that you have a smooth, invertible actually relationship between what happens in the sensory space and cortical surface. This is also true if you have very high resolution data for visual orientation. You see that in a given location, different orientations are mapped in neighboring neuron populations. And this can, to some extent, be seen with fMRI. And there is some continuity in this mapping. This is also the case for tonotopy. If you study the auditory cortex, you will see that some voxels are more responsive to some frequencies of the audio content, and there is a continuous mapping between the frequencies and the location of the voxels in the auditory cortex. It's actually hypothesized that it's also the case for the representation of numbers with numerotopy, where the one-dimensional structure of the number line that represents the quantities organized in a topological line, this might correspond to a one-dimensional structure on the cortex. This was the first intuition. The second one is that for a complex stimulus, you will have different levels of representations, more or less abstract. You can always characterize an image by relatively low-level features, like the local contrast in the image, the color histogram, or by more high-level features, like the semantics of the image. Oh, there's a face, there's a landscape, or something like that. And this corresponds to different levels of abstraction, and these different levels probably correspond to different brain areas. Okay? Oh yeah, normally this is a movie and it moves, but here I put a PDF so you don't see the retinotopic stimuli moving, but the wedge stimulus should be sweeping the visual field, and the ring should be a ring that you see expanding in the visual field. So this kind of stimuli are used to map retinotopic information on the cortical surface. This means that for a population of voxel in the early visual cortex, when you present these stimuli, so these stimuli are periodic, so you see that the voxel will have a strong response at the frequency of the stimuli. And so this is represented in that way. So this is the time course of activity in a given voxel in the early visual areas when we show the stimuli. And so all the voxels respond at the same frequency, but there are some phase differences. And the phase information corresponds here, for instance, to the location of the wedge in the visual field. For instance, the vertical position is a certain phase, while the horizontal left position would be a phase in quadrature, and so on. So when we map the phase information with the wedge stimulus, we obtain this map on the V1 visual cortex. Here we have drawn V1 on the cortex. And you see a smooth mapping from the lower meridian to the upper meridian. So where the left part of the visual field is represented on the right visual cortex. For with the ring stimulus, you would map the retinotopic information, whether the voxel responds to more central or peripheral visual information, and you see a continuous mapping from center to periphery with a much larger representation of the central part of the visual field. Okay, this is super well known. This has been done hundreds of times with fMRI. It was actually the first big experiment, big encoding experiment of fMRI was precisely retinotopy historically. And it's very important actually because it's the basic gradient to map the boundaries of visual areas in the human cortex. So we can map visual areas in the dorsal pathway, so starting from the occipital regions, going up to the parietal. So if we expand it, you go through V1, V2 dorsal, V3A, V3B, and then the intraparietal sulcus regions. And you see here, this is the wedge information, so the polar angle information of the visual field. You see how it is mapped along these regions. And same thing for the ventral visual cortex, going from the occipital to the temporal regions. You've got different visual regions, V1, V2 ventral, V3 ventral, V4, and then ventral occipital regions. And you see how the polar angle information is mapped on these regions. And similarly, how the eccentricity information is mapped on these regions. Okay, so this is a key experiment in which we really see some functional structure on the cortical surface through fMRI. Okay, to go a bit further, then we need to introduce the notion of model comparison. Because typically what will happen is that you will use some set of stimuli, and then you can generate different representations. So the so-called mapping phi that I called phi from the beginning, actually there's no unique choice of phi. You can create different features. For instance, from visual images, you could have features that characterize more the color content of the images, or the semantic content of the images, or the contrast in the spatial domain. So all these features could be extracted by different models. And so for the different stimuli, so different samples of your experiments, you generate these different features which corresponds to your encoding model, the phi. And then you will then see how well this phi1 representation explains brain activity in different locations using the R-square statistics, which I introduced previously. You can do that for the phi1, and then you can do that for phi2, and then compare in which brain region phi1 outperforms phi2. Phi1 outperforms phi2. So this is model comparison. So model comparison means in a given brain location, you see which model among phi1 and phi2 gets the best fit of the data. Okay? And so now with this, we can work on more advanced computational models of vision using the so-called framework of feed-forward architectures, in which a given stimulus is processed in different steps in order to achieve some representations which are useful, for instance, for object identification. So these architectures have become very popular in the last 10 years, but they had been there for 50 years, I would say, since Hubble and Wiesel discovery of complex and simple cells in V1. And so the idea, the principle has remained the same for now 50 years, which is that you can see the processing of visual activity as a sequence of layers in which a neuron will take some inputs, combine them linearly, and then will give the information to, will essentially apply some nonlinearity to this input and send it to the next layer. So during many years, the HMax model was the reference implementation for this kind of processing. So actually, the model was well specified for the first layer. It was less clear how the next layers should be implemented. So there were some implementations of this that were kind of working, but we were still lacking some principles to get good models for vision. So Stéphane Mala has introduced the scattering transform a bit more than 5 years ago now, which was a way using unsupervised learning to generate some this kind of representation. So the idea was really to cascade some kind of wavelet transforms of the input interleaved with nonlinearities. But it does not work that well. What really works in vision problems is the so-called convolutional networks model. So these models are artificial models designed to perform object recognition. They are exactly based on this principle, but simply the connections between the neurons are optimized to achieve the best object identification. So it's a supervised learning. Okay? So in the last 5 years, many such convolutional networks have become available. And so we have used them to see if they were a good candidate model to explain brain activity in humans. So for a collection of images that were presented to the participants, we have thrown the images into pre-trained convolutional networks. And so we have creating the phi of x, where each phi is the activity in a given layer of the network that comes when you present x. So it's the artificial neuron activity, if you like. And so for each layer, then you try to see whether it can explain the activity in each brain location using the so-called R-square statistics. And so for each layer of the convnet, you obtain an activation image that tells you how well each brain locations is explained by the representation generated by the layer 1, layer 2, etc., till layer 10, for instance, if there are 10 layers. And what you see is that actually the layer 1 will explain very well activation in the medial visual cortex, so in this part of the cortex, so in V1 and V2, while the fifth layer typically will explain less well V1 and V2, but a bit better the surrounding visual regions. And the final layer of the artificial network will not explain well V1 and V2, but explain well activation in the lateral visual areas of the cortex. Actually, it's a very consistent and beautiful mapping in which you can label each voxel by the layer of the convolutional network that best explains it. So for V1 and V2, it would be the first layer, and then the surrounding regions like V3, it will be the second layer, etc. And you've got this onion structure of the visual cortex that is recovered by that corresponds to the layers of the convolutional networks. And we can do that also by summarizing the activity in different V1. So when we know the borders of the different retinotopic regions, we can show that, for instance, V1 is best explained by layer 1 and then quite well by layer 2 and less well by the next layers. V2 is explained well by layer 2 and layer 1 and then less well. V3 is best explained by layer 2 but not so much by layer 1 and then decays and so on. So you really recover the visual region hierarchy by comparing the bold activation to the representation created by the convolutional network. So this has been observed and published by many groups since 2014. Yeah.
How do we determine how many layers we choose?
This has varied a lot, and I think it's still not fixed. Now you've got model with more than 100 layers, so ResNet used for object recognition in artificial vision. Of course, a biologically plausible model should have 5, 6 layers, something like that. So yeah, of course, there remains a tension between the optimization of the artificial network for a given task and what the biology has given us. Certainly, these convolutional networks were inspired by biological networks. But now I think they have, at least most of these networks, they have become quite different. So there are some attempts currently to create some networks that are more primate-like or human-like. So the cornet network has been designed for that. So it's a work by the DiCarlo and colleagues. So very similar things has been done in the monkey cortex with electrophysiology. This is why what I'm presenting today is not completely tied to the bold imaging. Actually, the same business can be done with electric recordings in the monkey. So they obtained, for these types of images, they analyzed these images through the convolutional network, and they tried with the convolutional network to explain activity in different locations of the inferotemporal cortex of the monkey. And what they found is that, well, some of the neurons in IT were very object specific. For instance, some neurons are chair neurons. They respond when the monkey sees a chair object, or there are some faces neurons which respond selectively to faces. Here is one example, for instance. And a bit as for humans, we could not, we really need these complex multi-layer convolutional networks to explain the activity in the inferotemporal regions. So the so-called HMO layer in that case. The old classical models like HMAX, which I presented earlier, were not very good at decoding. Okay, I'm going to skip that for the interest of time, but you can do the same business for auditory cortex, okay? I'll leave the slides available, so if you want to look more into that, that will be fine. So you can map, you can have a convolutional network for sounds, and you will see some regions that respond better to the first layers of the convolutional networks in red, and some regions that respond better, that are better modeled, better encoded with the last layers of the convolutional network. Critique of encoding models. This is interesting. I think there is some value in these models, but there are also some limitations. Maybe the first one is a lack of interpretability. Because typically, I told you that we estimate a beta in each voxel, which says how strongly each stimulus feature explains activity in this voxel. But actually, we don't look at beta because beta is very high dimensional. So how to look at that? So what we look at typically are the so-called R-square statistics. So we have these very high dimensional representations, and we summarize everything in a R-square statistic. So this is not very easy to interpret. I mean, there remains clearly some ambiguity in the interpretation of the model. I think this was partly related to your question. Second, model comparison is challenging because the data have a limited SNR. If you just repeat the experiments twice, of course you hope, like in a second session, you hope to recover similar things. But, well, there's lots of noise in the data which will not be reproduced. And actually, the best R-square that you can achieve is probably something like 0.5. Okay, so the true scale of values that you can achieve is between 0 and 0.5. And so this makes model comparison a bit challenging. So this means that you need a lot of data to be able to go to a clear conclusion on whether encoding model 1 is better than encoding model 2. You literally need to present thousands of stimuli to your participants. No mechanistic insights. Yeah, there's a little danger here. You could, for instance, use as features of the images the color in a given location of the visual space, and just encode it as RGB values. Okay, plain encoding of color information, and maybe this will explain activity in some given voxel. this doesn't mean that the neurons in these voxels are encoding the RGB channel information. This just means that they are sensitive to color. But maybe these neurons would be more sensitive to the saturation of the color. And if they are sensitive to saturation, of course, they will be explained a bit by the RGB values, but they will not be neurons that encode RGB values. So be very cautious when interpreting these encoding results. And since we always have limited stimuli because of time constraints, budget constraints, there can always be some confounding because objects tend to co-occur together. So a football ball will be always on a green background like grass. So if you can, the representation of this object will be correlated with other features of the images. So beware that the images have the features of images have a complex correlation structure, and the occurrence of object is maybe related to these features. And so it's very hard to avoid this confounding because it's present actually in the stimuli that we use. Okay, I only have 10 minutes left, I'm afraid. I will need to speed up a little bit. I will leave you the slides. This is an experiment that we ran recently on videos. Instead of working simply with still images, you could present a movie to the participant and try to encode this visual stream and see how well it explains activity in the visual cortex, in the cortex. And so here we took two such networks, one that was based on the optical flow, so the difference between successive frames, and one simply based on the RGB, so the color information across all pixels, so like a static model, but sampled in time. And we have different layers for each of these networks, and with this we could see in several subjects characterize the difference of the response of different voxels in the brain. We actually acquired data on the whole brain, but you see that the responses are strong only in the occipito-temporal junction and all the occipital regions, the middle part, etc. The interesting thing is that when we see which layer explains well which voxels, we obtain some relatively clear boundaries. Some of them are related to the different visual areas. Some of them are not, and it was a bit of a strange result. For instance, we saw, this is a contrast map that shows for each voxel whether it's best explained by the RGB visual network or the optical flow network. So RGB is in color, or in a yellow-red color, and the optical flow is in blue. And this contrast does not correspond to boundaries between visual areas. It actually corresponds to the representation of eccentricity in this subject. So we also did a retinotopy experiment in the same subject, and you see that the most peripheric regions in terms of retinotopy are those that respond to the optical flow in blue. And I think, sorry, I was wrong in my explanation. So the peripheral corresponds to the optical flow here, while the most foveal regions that see the central part of visual field are best explained by the RGB network. And actually, there is a very good correspondence between these two maps, which were acquired independently on the same subject. Yeah.
Did they fixate on the center?
Yeah. It was tedious for them. The subjects hate this protocol. I'm putting that because this data will be made available soon in the next months in the HBP knowledge graph, so they will be accessible to the community. it's part of the so-called individual brain charting protocol. So there's a web interface. So anybody can create an account, even if you're not part of the HBP yourself. You can create an account to get access to the public data, and this data will be public. And you can query data across species, protocols, and modalities. So here it's fMRI, but you can get it's fMRI on humans on these video watching protocols or retinotopic protocols, but you could query data from different species and protocols. And actually, yeah, HBP, Human Brain Project, has put a lot of effort into the data integration in the so-called neuroinformatics platform, where people can contribute data that are curated and integrated into what the so-called knowledge graph that links the data to metadata, which is important to be able to relate to them. Here, I'm referring only to the human fMRI stuff, but there will be some data on different animals, etc. And you can then query this data by either some meta information or the brain locations they relate to, and so on.
Which movie was it?
I think in that one we reproduced the one by Jack Gallant from Berkeley. So it's a short video clips. But recently we've been doing Raiders of the Lost Ark, which was much better for the participants. And we will be doing the good, the bad, and the evil soon. So it's an experiment in which we have selected a few subjects and we scan them 50 times, varying as much as possible the experimental conditions. Yeah, this is funny, so you could stop with that. One way to validate, I'm obsessed by validation, okay? It's good to tell nice stories about the brain, but it's even much more important to validate them. So how can we validate all that business? So one possibility is to try and reconstruct the images that were seen by the subject. And this is the converse operation, it's decoding. So you give me the fMRI map, and I try to predict what was seen by the subject. And so the thing works that way. You have the training images, you have the corresponding fMRI activity. With a generator, you can try and reconstruct the training image. And you've got, so the generator is a deep network, like a convolutional network that we talked about. And then you've got two networks, a discriminator that tries to predict if the image is real or fake. You want to reduce the gap between the true images and the fake images or the reconstructed images. So as in a GAN, generative adversarial network. And you also have a comparator which tries to make the reconstructed images as close as possible to the training image. And then this is at training time, so on a bunch of images. And then at test time, you take a test image, and based only on fMRI activity, you generate a corresponding reconstruction, which is not exceptionally nice in that case, but at least fMRI activity has captured something in terms of color or position of the input image. And so on. So this is demonstrated in this paper. So as you can see, it's kind of successful. I mean, it's not great. Obviously, it does not reconstruct the images so well. It usually captures the visual layout of the object in the image, for sure. Sometimes it captures a bit of color, a bit of texture of the images, and of course, quite well in the central part of the image where the subject fixates, and not so well in the periphery. Even more challenging, and this has been done by somebody from the HBP, so the team of Rainer Goebel. It's about mental imagery, where subjects were first perceiving some letters displayed on the screen, and you can reconstruct the image of the letter based on fMRI activity for the HTSC thing. So it works quite well in perception. And it works to some extent when the subjects no longer perceive the image, but now try to visualize by mental imagery, try to recreate the image of the H, the T, the S, or the C. Actually, funnily, the T works quite well, maybe because there is this vertical bar in the middle, which can be captured actually by the model quite well. It works not so well for all features of all letters. Actually, I had done an attempt at that a few years ago, and it's very challenging. But it turns out that we can, to some extent, and at least better than chance, generalize perceived images to mental imagery. Okay, it's time to conclude. So the name of the game was to compare complex representations of cognitive content to brain activity, and I think that the key concept is representations. Now with deep learning solutions, we have a great tool to create good representations of objects and benchmark them against brain activity. And this representations concept is very key in current deep learning technology. There's a big bottleneck, which is a limited SNR of the data. It's true for fMRI, but it's also true for MEG, EEG, or any modality that you would use. And because we can only acquire a limited number of samples, so there are lots of ongoing work. I think one of the interesting works, actually, it relates to your question. You can design different architectures for different tasks, and how well these different architectures will explain brain activity, I think it's a key question. And also working with language, for instance, is a big challenge, because language is a complex content for which now we start to have good computational models, and so we could compare these models to brain activity. Lots of resources, so of course go to the HBP knowledge graph and you will see some data, but there are also some public data, so you can look at them. We provide open source software which can be installed, and you can run the examples. I can guarantee that all the examples run, so don't hesitate. I'll be happy to help on that. And that's it. Thank you for your attention.